Okay. Does that sound right? Did we end up with talking, didn't we finish with MHC? Yes, could somebody give me a thumbs up? Not watched the thumbs up.
No, I
Okay.
don't. Did we go over that?
Does everybody want me to start with MHC again? Because
I
I know.
think that would be a good idea to go over it.
Okay. Okay.
Yes,
Let's
please.
go. It's a source of confusion for a lot of people. So let's start with MHC again. Okay. So MHC. So MHC, a lot of people have heard of the term HLA, right? Especially if you're someone who's ever read about like bone marrow transplants, for example, and you'll hear when people are discussing bone marrow transplants that they're looking for donors that are going to have HLA compatibility with recipients, right? So HLA, that's an acronym that stands for human leukocyte antigen, all right? That is just another word for MHC. So MHC is a type of HLA. And I bring that up because I think it helps people to visualize and to remember that MHC is it's a molecule that is expressed. Well, first of all, there are two types of MHC. And the acronym MHC stands for major histocompatibility complex, right? And MHC, there are two types of MHC. Both of them are specific to you as an individual, right? The first type of MHC is what we call MHC1. And MHC1 is an antigen or complex that is found on the surface of all of the nucleated cells in your body, right? That's why it's called HLA also, because it really is an antigen. It's a type of self-antigen, right? And what MHC1 does, and again, found on the surface of all nucleated cells of your body, but what MHC1 does is it processes and then it presents proteins or fragments that come from inside the cell. Okay, so MHC1 in a normal, healthy cell, nucleated cell, is going to be displaying self-antigen, right? Self-antigen are going to be fragments, protein fragments from within the cell, your cell, that lets your immune cells know that cell is you. It's what we call a native cell, okay? And then the other type of MHC molecule that we have is a molecule that's called MHC2. And just because I feel like with this kind of material that semantics can kind of trip people up, if you see MHC class 1, that's the same thing as MHC1. If you see MHC class 2, that's the same thing as MHC2. So MHC2 or class 2 is a molecule that is found on the surface of only 3 different cell types, okay? And the 3 different cells that have MHC class 2, they are all what we call professional antigen presenting cells, right? In this textbook, they will kind of just sometimes call them antigen presenting cells, but truly they are all cells that are nucleated are technically presenting antigen in MHC1, right? Self-antigen or antigen that comes from within the cell. MHC class 2, you only find on the surface of professional antigen presenting cells. Those cells specifically are B cells, cells called dendritic cells, and macrophages. And macrophages are the same cell as monocytes. They're called macrophages when they are in the tissue, and they're called monocytes in the blood, right? So those are your 3 professional antigen presenting cells that present antigen that comes from the outside, right? So these cells process and present antigen that they take in from the outside, right? And present it in MHC2, okay? So anything that comes from the outside is going to likely not be self-antigen, right? Because it's going to be something that the cell took in from antigen that the cell engulfed and took in, processed, and then presented. So how this relates to the T cells is that so T cells, right? The two main types of T cells that we have discussed have been what we call helper T cells, and those cells are CD4 positive. They're called CD4 positive. Oh, does someone have a question?
Did you go on the lecture right
Oh,
now?
somebody needs to mute themselves. Sorry, guys, I don't know how to mute upon entry after the fact. Okay. So, T helper cells, again, they're called CD4 positive T helper cells. And the reason they're called CD4 positive is because they have a molecule on the surface of their cells that's called, and CD stands for cluster of differentiation. And that molecule, that CD4 molecule, what it does is it stabilizes the interaction of the T helper cell with the antigen that's being presented to it in the context of MHC, okay? And I know that's kind of a mouthful, so let me use this picture to illustrate. So T cells and B cells, right? In the last lecture, we were talking about how T cells and B cells recognize specific antigen, because that is what makes these cells that are cells of the adaptive immune response, that's what makes the adaptive immune response special, is that it is the cells that make up the adaptive immune system are able to recognize specific antigen. They're able to mount a defense against a specific marker for a particular pathogen, let's say, right? So in order to recognize that specific antigen, they have to have receptors to recognize it. A B cell has a B cell receptor, which is just an antibody stuck, specifically what we call IgD, stuck to the surface of that B cell, right? And antibodies, and I'll show you some pictures and we'll talk about this in more detail here in just a moment. Antibodies can recognize free antigen. So antigen that does not have to be presented in MHC. Antibodies are able to just bind onto free antigen, right? T cells also have a T cell receptor to recognize their specific antigen, but T cells cannot recognize free antigen. T cells have to recognize antigen in the context of an MHC molecule. In other words, or otherwise said, T cells can only see antigen that is being cradled or held by MHC, okay? And that's the T cell receptor. The T cell receptor has to see the antigen being held in the pocket of an MHC molecule, all right? And then what the CD molecule does is it stabilizes the interaction between the T cell receptor and the antigen it's designed to recognize that's being held by MHC, okay? If we're talking about T helper cells, which are CD4 positive, circling back to that, CD4 positive T helper cells recognize the antigen they're designed to recognize in the context of MHC class 2, okay? Cytotoxic T cells, if somebody is unmuted, please everybody check to make sure you're muted. Cytotoxic T cells are what we call CD8 positive cells, okay? Cytotoxic T cells recognize their antigen via their T cell receptor in the context of MHC class 1. And then for cytotoxic T cells, the CD8 molecule stabilizes that interaction. So that's what you're seeing here, right? Here is a T helper cell. You know it's a T helper cell because it has this CD4 molecule that is stabilizing. Here's the T cell receptor. Here's the CD4 molecule stabilizing the interaction by which the T cell receptor is recognizing and binding to the antigen it's designed to recognize that is being held by MHC class 2, okay? In this picture here, you're seeing the cytotoxic T cell. You know it's a cytotoxic T cell because it has CD8 on its surface. And this CD8 molecule is stabilizing the interaction by which the T cell receptor on the cytotoxic T cell is recognizing its antigen in the context of MHC class 1. Okay? So what are the implications of this? So what this tells you, again, remember, and if I were a student and as I was learning this material, I think pictures are really, really helpful. You can draw yourself some little crude drawings to help yourself remember these interactions. But first and foremost, you need to remember that all nucleated cells have MHC class 1, and that MHC class 1, what that holds and presents is what is antigen that comes from inside of the cell. As such, we call this antigen endogenous antigen, okay? So MHC class 1 holds endogenous antigen, and it presents endogenous antigen to CD8 positive cytotoxic T cells, okay? So again, what kind of antigen, what are examples of endogenous antigen? Well, of course, self-antigens are endogenous. They come from within the cell. Another kind of antigen that is endogenous is viral antigen. Because if you can remember the way in which viruses work, viruses get into our cells and they hijack the host cell's machinery to make copies of themselves, right? So antigen, they get in and they insert either their DNA into our DNA, or if it's an RNA virus, they get into the cell and they use the ribosomes in our cells to make copies of themselves in viral proteins. In either scenario, if it's a virus, it's coming from within the cell, so it's endogenous antigen. So what that tells you is that cytotoxic T cells play a really important role in monitoring to make sure that the cells that are presenting antigen are cells that are presenting self-antigen, right? And if it's self-antigen, the cytotoxic T cell leaves the cell alone. It also tells you that cytotoxic T cells play a really important role in recognizing and mounting a defense against viral antigen, right? Because if one of your self cells is infected with a virus, that antigen is going to be presented to the cytotoxic T cells in the context of MHC class 1, because it's the MHC1 that holds endogenous antigen. Okay? And then MHC2, again, is only found on the surface of professional antigen presenting cells, B cells, dendritic cells, and macrophages and monocytes. Dendritic cells and macrophages, monocytes are phagocytic cells that go around and they gobble up debris, and then they present fragments of that debris. They process and present fragments of that debris in the context of MHC class 2. B cells, the way they work as antigen presenting cells is that when I was talking a moment ago about how antibodies are capable of recognizing soluble or free antigen, right? And that the B cell receptor is an antibody stuck on the surface of a B cell. If that B cell that has the B cell receptor for its particular antigen finds its antigen and its antigen binds onto that antibody, which is a B cell receptor, that initiates a phenomenon by which the B cell basically engulfs the antibody, the B cell receptor. It engulfs that receptor that's holding the antigen that bound to it, and it processes that antigen, and then it presents it in the context of MHC class 2 on the surface, right? And again, the cells that see antigen in the context of MHC class 2 are T helper cells. And cytotoxic T cells work. The cytotoxic part tells you that when they see foreign antigen, right, in the context of MHC class 1, they target and destroy that cell. T helper cells, if they recognize antigen in the context of MHC2 that's foreign, they don't directly kill the cell. They release cytokines that activate the immune response against the specific antigen that has activated that T helper cell, right? So again, exogenous antigens presented by MHC2, endogenous, coming from within the cell, presented an MHC class 1. And that's what these slides go over. And as I mentioned, I feel like pictures are really useful when it comes to this and trying to sort out the moving parts involved in these processes. Okay, and then when it comes to organ transplant and the role that MHC molecules play in organ transplant, the beginning of this lecture, I was using the example of a bone marrow transplant and an HLA, right? And I told you that HLA is just another name for MHC. So HLA / MHC, these molecules are, again, they are self-antigen molecules, right? That's why they're called HLAs, human leukocyte antigen, as they are specific to you as an individual, right? And so they play a really important role in when we talk about tissue transplant, whether it be blood or bone marrow in the case of a bone marrow transplant, or an actual organ like a liver transplant. There's this idea of compatibility becomes really important. We try and get as close a match, and what that means to be a match is that the molecular pattern that makes up your MHC molecule, if you are the donor, that they're going to try and find a recipient, somebody who needs, a kidney. If you're donating a kidney, they're going to try and find a recipient whose MHC molecules look as close as possible to yours, because the closer the molecular match, the less likely rejection, the lesser the likelihood of rejection, right? The farther away or the more dissimilar the molecular pattern of MHC molecules between recipient and donor, the more likely that tissue will be rejected. So the only time that you don't really worry about this would, of course, be an identical twin. If you had an identical twin and you needed a kidney, you wouldn't have to worry about compatibility because your MHC molecules would be identical. If you don't have an identical twin, and most of us don't, the next best thing is to try and oftentimes those matches, of course, you're statistically speaking, you're more likely to find those matches familially speaking. So family members are going to be more likely to have those MHC molecules that are molecularly speaking closer to yours compared to a stranger, but that's not always the case. That's why they have donor lists where you put yourself on a list, and they will test your MHC to see how closely related it is to any of the potential or people who need the potential recipients out there who are needing a tissue. And oftentimes you find a match between people who aren't even in the same country. But that's this whole idea of organ transplant in MHC, is that you try and get that match as close as possible in order to avoid what we call graft rejections. Okay, so now we're going to go through and we're going to run through the life events for B cells and T cells. And we're going to kind of use T cells to highlight many of these life events, right? So most of this information initially is by way of review, right? We already know, because when we were studying the lymphatic system, we already know that B cells and T cells arise in the marrow, right? And we also know that T cells, after they are initially formed or originally formed in the marrow, they immediately migrate to the thymus to mature, right? As such, both the bone marrow, red bone marrow, and the thymus are what we call primary lymphatic structures, right? Because they are where these lymphocytes originate. And what happens once and we're going to go through this, and I'm going to bring up these terms now and then talk about them more in a moment. there's this difference between, or there are two terms that we use to describe lymphocytes after they are formed. And the two terms are naive versus, well, I should say naive and then immunocompetent, okay? So what it means to be immunocompetent for a T cell or a B cell is that you as a T cell or a B cell are able, you have a receptor that allows you to recognize your specific antigen, and you are able to recognize it and not react. In addition to being able to recognize your antigen, you also do not react to self-antigen, okay? That's what an immunocompetent T cell or B cell is. This idea of being a naive T cell or B cell, if a cell is naive, it tells you that if it's a cell that's naive and immunocompetent, it tells you that it's a T cell or a B cell that can recognize its antigen, but it never has seen its antigen before, okay? So that's what, when we were in the lymphatic system, when we talked about lymphatic structures as being surveillance tissue, right? What you find in lymphatic structures, especially in someone who is very young, are a bunch of naive, immunocompetent but naive T cells and B cells, right? They're there, they're able to recognize their antigen, but they've never seen it yet. And that there are some important implications for that. If you have a naive T cell or B cell, those cells they've not ever been activated, so they have no populations of memory versus effector cells. They've never seen their antigen. Once a B cell or a T cell has seen the antigen they're designed to recognize, they will undergo activation and they form memory cells, right? And memory is a very, very important concept related to the adaptive immune response. It belies this whole phenomenon by which if you get infected with a particular virus and you get sick, or you're vaccinated, which we'll talk about in the third part of this presentation, and then you see that same virus another time down the road, you have such a fast response against that virus, that's such an antigen specific response, that you don't even get sick, right? And that's because you have memory cells. There's memory that exists for the adaptive immune system. So like I said, and that here are those words here, naive and immunocompetent, right? So you have, when cells leave, I'm gonna minimize that. When B cells leave the red marrow, they're going to be naive and immunocompetent, and they're going to migrate to the secondary lymphatic structures to await exposure to their antigen, right? T cells that leave the thymus, same thing. They're naive and they're immunocompetent, and they also, like the B cells, are going to migrate to secondary lymphatic structures to wait exposure to their antigen. Okay, so that's what this slide goes over, right? The activation of T cells and B cells takes place almost entirely in lymphatic structures, in lymphatic structures, and also, in circulation, but surveillance via, let's say, lymphatic nodes or lymph nodes is incredibly important when it comes to activation of T cells and B cells. And the T cells and B cells that are hanging out in secondary lymphatic structures are going to be naive and immunocompetent, right? And then what happens is that, let's say we have a B cell, for example, that comes into contact with its antigen, right? And I guess I should also mention the reason the lymphatic structures are so handy when it comes to activation of lymphocytes is that if you think about, again, lymph is formed as plasma, or as blood moves through capillaries, and essentially plasma and other components within plasma, which would include viral antigens, and even smaller protein cellular debris to some degree, but definitely viral antigens. Those get pushed out into the interstitial fluid and then get pushed into the lymphatic vessels to become lymph. So if you have in that scenario, if you have a B cell waiting in a lymph node that comes into contact with let's say, a viral antigen, it will bind to that B cell receptor and it will activate that B cell receptor. And what will happen through that activation process is that that B cell will become activated into one of two different cell types. It'll be activated to become what we call the effector cell. And in the case of a B cell, the effector cell is what we call a plasma cell. And what that plasma cell does, or what they do, is they just churn out a bunch of antibodies that are designed to recognize the antigen that activated that B cell. Okay. And those antibodies are soluble. So the B cells themselves, as they become activated, they hang out in the lymph node or the lymphatic structure in which they were activated. And the effector cells, the plasma cells, just start to churn out huge quantities of neutralizing antibodies that get into the circulation and travel around and bind up, recognize and attach to the antigen that activated that original B cell that again became activated and differentiated into a plasma cell. And we'll talk about the different ways in which antibodies work here in just a moment. And then T cells are a bit different in that when T cells, so if you have a T cell hanging out in a lymph node that becomes activated, T cells become activated, they start to undergo proliferation. So the effector cells start to undergo mitosis to make a bunch of copies of themselves. And unlike the B cells, the T cells don't continue to just hang out in the secondary lymphatic structures. They actually get into circulation, and they circulate around, and they also migrate into the tissues. They follow chemical trails, and they migrate into the tissues where the site of infection has occurred. Okay. So now we're just going to kind of hone in on the formation specifically of T cells. So when T cells, so we're going to kind of backtrack here a minute because we just kind of ended talking about the activation of B cells by their antigen. But we're going to back up a moment and talk about the specific mechanisms by which within the thymus, T cells are made to be immunocompetent and naive, right? How they are able when they leave to the thymus to be immunocompetent and naive. So what happens with them, again, T cells, the original precursor, if you will, T cell is made in the red marrow, and then those cells very quickly migrate to the thymus. And when they get to the thymus, these precursor T cells have both, so they're not functional T cells, they have both CD4 and CD8 on their surface. And each one that migrates there has a unique, and I should say this goes the same for B cells, has a unique T cell receptor, right? The way in which T cells and B cells, and the fact that T cells and B cells have almost an infinite number of unique receptors for an infinite number of unique antigens, that all takes place through gene rearrangement and processes that are beyond the scope of this class. You'll talk about them if you take immunology, but just suffice it to say that each one of the receptors that you find in this particular context on a T cell, each one of those T cell receptors is going to be unique, right? And designed to recognize one and only one type of antigen. And so then what happens within the thymus is this process by which the T cells, those pre-T cells, precursor T cells, are tested to make sure that their T cell receptor is able to recognize the antigen that receptor is designed to recognize, and at the same time to ensure that that T cell receptor does not recognize self-antigen as being foreign, right? Because if you end up with cells that T cell receptors that are functional and recognize antigen, but ones that that start to see self-antigen as being foreign, right, that are activated by self-antigen, that's when you run into problems with autoimmunity, right, and making cells that are autoreactive. You do not want T cell receptors or B cell receptors, for that matter, to see self-antigen as being foreign. You don't want self-antigen to activate T cell receptors or B cell receptors. So within the thymus, what happens is that these cells get tested. That's what positive and negative selection are. Tested to make sure that their T cell receptors are able to recognize and bind to the antigen they're designed to recognize and bind to, and at the same time ignore self-antigen. And then they also selectively will lose either CD4 or CD8. And so when the T cells leave the thymus, they're either going to have CD4 or CD8, so they're either going to be cytotoxic or T helper cells, and they're going to have T cell receptors that recognize their antigen and ignore self-antigen, right? And once those two things are accomplished, that's when you have a T cell that is immunocompetent, right? So here is that process. Here is the slide that goes over those processes of negative and positive selection, right? And I should say that this selection process gets rid of all of the pre-T cells that enter the thymus. Only roughly 2% make it out, if you will. 98% of the T cells, pre-T cells that enter your thymus are eliminated. And they're eliminated because primarily they're eliminated because they don't pass negative or positive selection, right? So what happens, and here's what these processes entail. So with positive selection, again, we're talking about pre-T cells. They have at this point both CD4 and CD8, right? And during positive selection, the cells that are selected for are going to be those cells that are able to recognize, or are going to be able to recognize their antigen in the context of MHC, right? So because again, that's how T cells see antigen. They see their antigen in the context of MHC. So that the T cell receptors are tested with, it just so happens, I'm not going to ask you, but this much detail, but these are thymic epithelial cells that present antigen in the context of MHC to the pre-T cells. These epithelial cells are going to test out both MHC class 2 and MHC class 1 to MHC2 to the CD4 cells. Well, they have both receptors at this point, but cells that are pre-T cells that are able to recognize their antigen in the context of MHC class 2 will become CD4 positive. Those that are able to recognize their antigen in the context of MHC1 will become CD8 positive. But the process of positive selection is testing those T cell receptors to ensure that they are able to recognize their antigen in the context of MHC, right? If they are not able to recognize, if they're not able to bind MHC, which is how they will recognize their antigen, they are told to undergo self-destruction, right? They undergo apoptosis. If they are able to bind or to fit with MHC, and therefore they'll be able to recognize their antigen in the context of MHC, they are allowed, if you will, they survive the process, okay? And so for those that survive, they then go through what's called negative selection. And negative selection is the process by which cells that see self-antigen, that bind self-antigen too tightly, those cells are selected against. That's why it's called negative selection, right? So what happens during negative selection is the cells that made it through positive selection are then tested to make sure that when these cells come into contact with self-antigen in the context of MHC class 1, that they're able to not, and I always kind of use the analogy of a hug, right? But these T cells are able to make contact with the epithelial cells holding MHC class 1, able to make contact with it, but they aren't activated by it. They don't hug it, right? Because what happens if a cytotoxic T cell, for example, sees MHC class 1 holding foreign antigen, that T cell receptor kind of hugs the MHC molecule holding the antigen. And it's that hug or that fit that activates that cell, right? So what should happen if the MHC class 1 is holding self-antigen is that cell comes along and goes, boop, checks it out, and then leaves it alone, right? And the same goes for the CD4 cells with the MHC class 2. So it's both these cells that still at this point, the pre-T cells, are tested to make sure that they don't recognize self-antigen, that they don't, the interaction between the T cell and the cell, the thymic cell with the MHC class 1 or 2 on it, that interaction is transient. Because again, the longer the interaction and the tighter the hug, the tighter the fit, the more likely it is that those cells will be activated. And so what happens if the interaction goes on for too long, and or if the hug is too tight between the MHC holding antigen and the T cell receptor, those cells are selected against. They're told to undergo apoptosis. And if they do not, if they make a transient, if they have transient interaction with self-antigen, then those cells are selected for, because those cells will hopefully not be autoreactive. So those are the cells that are selected for. So it's the cells that make it through positive selection, right? So the cells that are selected for via positive selection, and then selected against via negative selection, that ultimately are going to make up that 2% of cells that leave the thymus, that at this point are, again, as soon as they make it through positive and negative selection, they are immunocompetent, but they are naive. And they're naive because they have not been activated by the antigen that they are designed to recognize and be activated by. Okay, so that's what this slide goes over. And then what happens is you get the last step of this process right before they leave the thymus is that you get this selective loss of either CD4 or CD8. If you lose CD8, you're a CD4 positive cell, so you're a T helper cell. If you lose CD4 and retain CD8, you're a cytotoxic T cell. Okay, and this is just a slide you can use to study positive and negative selection, and then again the last part being the loss of the CD8 or CD4, and at that point again then you have immunocompetent but naive T cells that are going to get into the circulation and travel to secondary lymphatic structures where they will await interaction or exposure to their antigen. Okay. So that's what this slide goes over right here. Again, these concepts or these definitions of immunocompetent versus naive, right? And then one other category of T cells I want to make mention of are what we call the regulatory T cells. So regulatory T cells are a special type of T cell that we're not going to talk a ton about. What I want you to know is that these cells are cells that are involved in helping to prevent autoimmunity, right? So they play, mechanistically speaking, a really important role in helping, they kind of inhibit the immune response, specifically in a way that helps to eliminate any kind of autoimmune type of reactions. Reaction. And they're, regulatory T cells play a really important role when it comes to, well, obviously, the disease processes involved in autoimmunity. They also play a role in oncology and the development of some cancers. There are some, some tumors are able to essentially hijack this the regulatory T cell function and help to kind of make the immune response quiescent, or kind of make the T cells, the CD8 and CD4 positive T cells, a little bit less alert, if you will, for lack of a better way to describe it, because I don't want to go into too much detail and confuse people. But like I said, I just want you to know that regulatory T cells are involved in tolerance and helping to, when homeostasis is maintained and they are functioning correctly, these cells help to ensure that that autoimmune reactions aren't likely. Okay, So this concept of clonal selection, right? I've already kind of touched on when I touched on the activation of B cells, right? So what happens when let's use a B cell as an example. What happens when you have an immunocompetent but naive B cell that's activated by its antigen is that activation causes, again, the effector response. A big part of that effector response is what we call clonal selection. So clonal selection is going to be the production of a bunch of, in this case, B cells that are exactly the same, right? And as such, all of those cells will have exactly the same because we're talking about B cells, B cell receptor, and that B cell receptor is going to be designed to recognize exactly the same antigen, right? And this kind of brings me, which I think has some particular relevance right now in the news, because if you've been watching the news, you've probably been hearing about Regeneron's monoclonal antibody cocktail that is still considered investigational. I think it's in phase 3 of the clinical trials, but it was given to Trump recently because he has COVID. And what monoclonal antibodies are, these are antibodies that come from monoclonal B cells. So that's what this idea of clonal selection is. It's a B cell that, in this particular context, a B cell whose B cell receptor is designed to recognize a spike protein on the coronavirus, on that capsid, one of the spike proteins. And then that one B cell has been cloned, right, has undergone clonal selection, albeit in a laboratory setting. And as such, all of the antibodies that are made from that clone are going to be exactly the same, right? That's why they're monoclonal antibodies. They're all exactly the same. They're all from genetically speaking, exactly the same plasma cell. Polyclonal antibodies are antibodies that can be slightly different because they come from different B cells, right? And kind of more on that to come right now. But that's just to kind of give some relevance to this particular part of the slide right there. So again, the first time, so when you have an immunocompetent but naive T cell or B cell, right? The first time that T cell or B cell is going to see its antigen is almost certainly going to be in a secondary lymphatic structure. Can be in the blood as well, but it'll more likely than not take place in a lymphatic structure. So what happens is that they, again, the antigen will bind to either the T cell or it'll be presented to a T cell in the context of MHC, or it'll bind directly to the B cell receptor, and it'll activate that cell. And we're going to use T cells to illustrate some of the processes that are involved in this activation process, right? And we're going to talk, and the process is, we're going to talk about T cells, B cells, and then we'll finish up talking about, T helper cells, cytotoxic T cells, and then we'll finish up talking about B cells. So here's how T helper cells, immunocompetent and naive T cells, here's what happens the first time they encounter their antigen. Pardon me. So what happens, let's just paint, or let's come up with a scenario. So we have a scenario in which a B cell, right, comes into contact using its B cell receptor with viral antigen. And again, the viral antigen will bind to the B cell receptor. The B cell receptor will engulf the B cell receptor that's bound to the antigen, and then it will process and present that antigen in the context of MHC class 2 on the surface, okay? Now let's say that cell that B cell with MHC class 2 holding the antigen comes into contact with a T helper cell that is designed, its T cell receptor is designed to recognize the antigen that's being held by MHC class 2 on the B cell. right? What happens is there is a physical interaction that takes place between the T helper cell that's naive but immunocompetent and the MHC class 2 holding the antigen on the surface of, in this case the picture I just painted, a B cell. And so they make physical contact with one another. The CD4 molecule again stabilizes that, that interaction. That's what you're seeing APC here. And then it stabilizes that interaction so that that interaction, again, it's that idea of a tight hug, right? The T cell receptor CD4 molecule hugs the antigen that's being held by the MHC class 2. And that hug can last, from, several seconds to several hours. The longer the duration of the hug, the more likely it is that that T cell is going to be activated, right? And then what happens if that hug goes on for an adequate amount of time, if you will, is that that T helper cell will start to secrete IL-2, which is interleukin 2, right? That's a cytokine. And I mentioned this in the last lecture, but T helper cells, again, they don't directly attack and destroy cells. What they do is they activate all of the branches of the immune system. They activate all of the other immune cells, and they do so through their very complex, extensive cytokine profiles. So one of those cytokine profiles is IL-2. So the IL-2 that is secreted after, again, this is the T helper cell that's been, that's recognized its antigen in the context of MHC2 on the professional antigen presenting cells, stabilized by CD4. It's gone on for a bit. And so this T helper cells become activated, starts to secrete IL-2. And IL-2, basically that's secreted by the cell, feeds back onto that cell, because this T helper cell, all T cells have receptors for IL-2. So the second signal, the first signal would be the T cell receptor recognizing and binding antigen presented in the context of MHC2 by the antigen presenting cell, and that interaction stabilized by CD4. And then the second signal would be the IL-2 that is secreted by this cell coming back and further activating the cell that secreted it, okay? And then there's another, what we call secondary signal. I'm sorry, not secondary signal. I'll call it it's a type of co-receptor signal that is incredibly important when it comes to the activation of T helper cells, okay? And that and I should say the co-receptor signal is different from the secondary signal. The secondary signal is the cytokine response and the cytokine binding onto the cell that secreted it in this scenario. The co-receptor signal involves two molecules. So one molecule is called B7. You find B7 on the surface of the professional antigen presenting cells. And B7 binds a molecule called CD28, which is found on the T cells, on the T helper cells. And so it's during this, again, going back to this first signal by which you have the T cell receptor presenting, the T cell receptor recognizing and binding its antigen in the context of MHC class 2 that's presenting it, right? And MHC2 in the antigen or on the antigen presenting cell, and then CD4 stabilizing it. And simultaneously, you have B7 interacting, and this B7 and CD28 are not drawn on these pictures, but you have B7 that you find on the surface of the antigen presenting, professional antigen presenting cell, interacting with CD28 on the T cell. And that that co-receptor signal is required in order for memory to take place, right? So what happens if you do not have this co-receptor signal, this interaction between B7 and CD28, if that does not exist, you can still get activation of the T cell, and you can still have that T cell secreting cytokine profiles, but you do not get a good, you don't get good formation of memory cells, right? because it's the two signals plus the co-receptor signal that together fully activate the T helper cell, right? Fully activate the T helper cell such that that T helper cell becomes activated so that it will differentiate into one population of cells that are going to be the effector cells, the cells that are actively involved in fighting that infection. And then another population of cells that are going to become memory cells that retain that specific T cell receptor. And so that anytime those memory cells encounter that same antigen in the future, the activation process is that much faster. So much faster, in fact, that you do not get, you typically don't even get sick, or hopefully you don't get sick. Okay. So I know that was kind of a verbose way to describe it. Like I said, I think pictures are so important. These processes make a lot more sense if you can kind of sketch them out, right? So for cytotoxic T cells, don't worry, we're not going to talk about any kind of co-receptor signal for cytotoxic T cells. So cytotoxic T cells, the first signal, just like what we saw with the helper T cells, is going to be direct contact between the T cell receptor of the T cell and then the MHC molecule. In this scenario, it's going to be MHC1 holding its antigen. So first signal is direct interaction between T cell receptor and MHC1 holding the antigen that T cell receptor is designed to recognize, stabilization of that interaction by CD8. And then the second signal for the cytotoxic T cell activation is going to be, remember that IL-2 we were talking about that was secreted by the helper T cell that was activated? That IL-2 is also going to act as the second signal to activate the cytotoxic T cells. So just like with the helper T cells, first signal is interaction of T cell receptor with MHC and stabilization by CD8 molecule. And then second signal is going to be the cytokine IL-2 binding to IL-2 receptors on the surface of the T cell. And for cytotoxic T cells, once these two events have occurred, the cytotoxic T cell becomes fully activated and differentiates into a population of activated effector cells that will go along and look for that same antigen being held in the context of MHC1 on other cells and destroy the cells that are holding it. And then also form a population of memory cells, which again, just like the T helper cells, if you are subsequently exposed to that same antigen, those cells will very quickly differentiate into effector cells so quickly that you hopefully won't even become symptomatic. Okay, and then for B cells, the big difference between B cells and T cells is, number one, there's no MHC when it comes to recognition of the antigen by the B cell receptor. The B cell receptor does not need that antigen to be presented to it in the context of MHC, right? B cell receptors can bind free antigen. So the first signal for B cell activation is going to be antigen binding to the B cell receptor, which, as I keep saying, is basically an antibody stuck on the surface of the B cell. And then what happens is, again, as I mentioned, that when that happens, that B cell will engulf that antigen antibody or B cell receptor antigen complex. And then in order for the B cell to become fully activated, the second signal is the binding of the cytokine IL-4 onto IL-4 receptors on the B cell surface of the B cell. IL-4 is another cytokine that is secreted by T helper cells, right? So here again, you see T helper cells being the orchestrators of the immune response via their cytokine profiles, right? So it's IL-4 that is the second signal for B cells. It is IL-2 that is the second signal for both helper T cells and cytotoxic T cells. And then in response, once you have the first and the second signal, this now activated B cell differentiates into, just like with what we were talking about with T cells, into two populations of cells. The effector cells, the cells that are going to actively fight the infection for B cells, are cells, pardon me, that we call plasma cells. As you can see in this picture, plasma cells morphologically look very different than the cells from which they arose, and also from memory cells. Plasma cells lose their B cell receptor because that's no longer required for a plasma cell, because a plasma cell is essentially an antibody making machine. That's what it does. This plasma cell is going to just churn out tons and tons of antibody, the same antibody that is designed to recognize the antigen that activated it initially. Okay, that's what plasma cells do, is they churn out antibody clones, right? So, and then going back to this idea of a monoclonal antibody, these are all going to be monoclonal antibodies, right? And then the memory cells, as far as their morphology is concerned, look a lot like the original T cell from which they arose, right? These memory cells have the B cell receptor that activated them on the surface, and it has many copies of that B cell receptor on the surface. So again, the next time you encounter the antigen that initially activated your B cell, you now have a whole host of B cells out there that have B cell receptors that are designed to recognize that antigen. So when you're exposed that secondary time, you will have a very robust, very fast response to that antigen. And these cells will very quickly differentiate into plasma cells that will churn out neutralizing antibody to that antigen. Okay. So that's what this slide goes over. Again, plasma cells are the cells that secrete antibodies. Memory cells are the cells that kind of wait in the wings. They stay in those secondary lymphatic structures. They hang out there and wait to see if they encounter that same antigen again. And then with regard to recirculation, so this is, I mean, I have it here as kind of like a footnote, but I just want to bring up the fact. So, you know, at the end of the day, what happens, again, I kind of left off talking about those memory B cells and memory T cells hanging out in secondary lymphatic structures. But I think it's important to note that periodically throughout the life cycle of an organism, what will happen is that one of those, I mean, I'm using one, but one or a handful of those memory T cells and B cells, they kind of leave, if you will, the secondary lymphatic structure, and they get into the circulation and they roam around just in case there's a chance encounter in the circulatory system with the antigen that they are designed to recognize and be activated by. So, and that happens throughout our lives. As long as you have memory cells, those memory cells, most of them are kind of waiting in the wings and those secondary lymphatic structures, but then they are constantly, you know, a few of them at a time getting into circulation, circulating around, looking for their antigen. And if they don't encounter their antigen, great, they undergo apoptosis, and that's it for that particular cell. And if they do encounter antigen, then they, of course, will be activated to differentiate into effector cells to defend you against that particular assault. Okay. So let me see. I think I said most of what's on this slide too. Again, helper T cells, kind of the take home is that helper T cells do not directly destroy cells, right? They use their cytokine profiles to activate immune cells and therefore orchestrate the immune response. We were just talking about IL-2, T helper cells secreting IL-2 that will both activate themselves, and also cytotoxic T cells, and IL-4 as being the cytokine that is the second signal that activates B cells. And then how cytotoxic T cells, unlike the T helper cells, cytotoxic T cells do directly destroy the cells that they recognize as being as holding foreign antigen. And then of course, plasma cells are activated B cells that are just antibody making machines. Okay, I think I said everything I want to say on this slide, again, helper T cells are called helper T cells because they help out the immune response. They help to activate all of the other immune cells that we have. And then cytotoxic T cells. Cytotoxic T cells, their exact mechanism of toxicity is very similar to that of a natural killer cell. So what they do when they recognize their antigen as being foreign in the context of MHC1 is they secrete what's called perforin, and perforin drills a hole into the cell that they recognize or that they've been activated against. And then into that hole, they insert what are called granzymes. And granzymes induce apoptosis or pre-programmed cell death in the targeted cell. Okay. And then when it comes to B cells and this concept of an antibody titer, so again, activated B cells that have become plasma cells are those are antibody making machines, right? And they're stationed, those plasma cells are stationed in the secondary lymphatic structures, but they're just churning out lots and lots of antibody against the particular antigen that activated that initial B cell that turned into a plasma cell. And what an antibody titer is, an antibody titer is a measure of the concentration, circulating concentration of an antibody against a specific antigen in the blood, right? So titers can be done for, titers are an important part of trying to gauge whether or not somebody has protection against a specific antigen, right? So for example, if you've been vaccinated against hepatitis B, and which is, at least when I initially got it, it was a series, I think of, 3 to 5 shots spaced a few months apart each. That regimen might have changed, but all the same, if you've been vaccinated against Hep B, what you can do if somebody goes in, you don't have your immunization records, and they say, well, have you been vaccinated against Hep B? And you say yes, and you don't have proof of it, they can do a titer. And what they do is they take your blood and they look for levels of circulating antibodies against the Hep B antigen in order to determine, are you immune? Or do you have neutralizing antibodies against this antigen circulating around? The assumption is if you have a robust enough titer, if you have a titer above a certain concentration, the assumption is that you are protected, for all intents and purposes, protected against that particular antigen. And this, I'm using Hep B as an example. So if you have a titer, a strong enough titer or high enough titer for the antibody against Hep B, the assumption is you are immune to Hep B, unless maybe your system becomes overwhelmed because you get a huge dose, huge viral load, or you become immunocompromised or something of that nature. But that's what titers are used to determine the concentration of circulating antibody against a particular antigen. Okay, so I am out of time. Let's see how far we got. 36. Okay, so I will have to record the rest of these slides and part 3, which is only 15 slides long. So I have about 25 slides to get through. I'll record the rest of that this afternoon and post it as a separate file. As you all know, this recording right now is recorded using Zoom. So you will find it in the Zoom cloud under today's date, the 5th of October. And then when I pre-record videos, I use Panopto. So I will just place that file. I'll make an announcement about it, and I'll put that file into the Zoom lecture recordings folder on Blackboard. And I'll do that tonight so that everybody has that to study with on the upcoming exam. Okay, so I'm going to stop this recording. And if you need to reach out to me, feel free to do that individually.